that we could culture or isolate DNA.
Okay. Makes me think of cookie dough.
Yeah, the sandy ones really were like those tubes of sugar cookie dough.
Right.
These ones are more like goop. I think this will be a much finer, muddier sediment.
Will they still retain the shape even though they're
That one's pretty good.
I feel like the finer ones, they kind of, I don't know, you guys have been dealing with them. Do they sort of pour out or do they keep their structure pretty well?
No, some do like kind of like melt into a more liquidy, I think, paste. It's really
but
nice.
I think I'm going to ask you to do one more core here, just as a backup.
That was
After this.
That is NA124172, push core 1.
So hold up there.
I hope there's nothing
So hold up.
in
What's
the
it doing?
quiver.
There it goes. Ooh, so strong.
You'll never get that cork off.
sample tray in.
So one more, please.
Yep.
poor Lisa's being torn in the directions of good sediments here and a possible seep related. Fingers crossed. There's a lot of finger crossing today.
Indeed.
Okay, I'll zoom in. Starfish free?
I think
Yes,
so.
please. Yep.
Seems
There.
okay.
That looks all
That
right.
looks fine.
You can see the dirt being pushed down in there.
Yeah.
So
Yeah,
maybe I'll kind of go a little deep on it.
Go deeper, yeah. That's good.
Look at that little section. It's so fine. Like a
Wow, that really compresses.
Yeah. Should have gone mow deeper.
It's okay.
all right, this will be push core 6.
Push core 6, yep. 173.
Yeah.
All right.
Thanks, Bob.
Thank you.
So what are we doing now?
Now, we wanted to slurp those 2 cucumbers up ahead.
Okay.
I think Greg called them a warty cucumber, and we would like to use either 4, 5, 6, section 4, 5, 6, or 7.
Sure.
Carly, you gotta go to flush because I think we got a lot of dirt in the hose here.
Okay. Let me get the flush jar on for you.
And for those tuning in, we call this a slurp. It's basically a little vacuum, I guess.
Little vacuum cleaner.
Ready
Yeah.
for flush?
Yep.
And they have one jar that they use just for getting stuff out of the hose. And we use it to collect biology and sediments, basically things that we'll want in a little jar to look at later that are hard to pick up with the ROV claw.
Ooh, look at all that dirt. Oh, man.
Porch full of mud.
I hope that'll fit. Will that big one fit?
Is
pretty
it
big.
cool if I turn off flush now?
Yeah.
Now that all that dust is flying around it. We want 4, 5, 6, or 7?
Yes.
Okay. It's like Mad Max. Dystopian, deserty.
Very deserty. I watched the whole series in quarantine.
Oh my gosh.
Yeah, it was
What
it's
did you watch?
the whole Mad Max series. It's been on like my to-do list forever. And in quarantine, I'm like, this is the time. And I had the big hotel screen and like the HDMI cable in, and I did the whole series.
That's awesome.
I'm totally caught up now.
Yeah, that's true. In prep for the cruise, everybody on board did a 2-week strict quarantine, either at home if they were able or if they traveled across the country like a lot of the OET staff did. They had to stay in a hotel room for 2 weeks.
Just for the pleasure of sitting here with us.
It was worth it.
I got to do it twice.
Oh,
Me
That's so
too.
good.
Me too. I think I may be the only person who didn't mind it though. Everybody else was miserable. I was like, this is fine. This is great.
Okay, Al, zoom in. We don't have full zoomage, but
You want suction now?
yeah. let me
Which
figure out-
canister are we going to?
It's 7.
7.
7?
Ready?
Sure.
Sure.
I hope he doesn't get stuck in
Oh,
there.
he looks like a tube
Yeah.
clogger. Oh.
Whoops.
No, I don't know. He went fast.
and that's why we call it a slurp, folks. He was slurped right up.
Please
Hopefully
make it
he
to the
slurps right through.
to the jar. Go to the jar.
Don't stick.
I can't tell if there's something under there or not.
Trying to come through. Yeah.
So this ended up in slurp jar 7?
Yes, almost.
It's on its way, maybe.
Unconfirmed. I wonder if the porch light's on and that's the glare.
Cycle it a couple times.
Okay.
That was porch light glare, so that was not cucumber. Lots of
Alright.
stuff.
Turn that off again and whip it back on.
Sometimes things get stuck on their way down the slurp tube. It reminds me in Willy Wonka, who was it that gets stuck?
Augustus Gloop.
Augustus Gloop gets stuck in the
Save some room for later.
chocolate tube. If this little cucumber would have just eaten a little less, could have fit right through.
Yeah. The other one's skinnier.
I'm gonna push him in, you know?
I don't know. I think he's stuck.
You want to try blowing them out?
Can't do that.
That'd be funny though. It's like a potato cannon.
That's a great visual.
You won't know what happened to him today.
We need a rotor-rooter thing.
There's a bit more stuff coming in. I'll cycle it again. I cycled a few times.
There he is.
Oh,
Yeah.
oh, I shouldn't have turned it off then. Okay, I'm back.
Wow.
Back, come on.
Wow.
That's a big guy.
Get in
Yeah.
there.
It is. I mean, dare we try the other one?
The other one does look a little smaller.
It's quite a bit skinnier.
They're going to be cozy in that jar together.
Well, they'll get along.
I still have suction on, just so you know,
Okay.
because he looks uncommitted to the jar.
Geez. It's very dirty here.
Uh-oh.
Yeah.
You want me to kill it?
Huh?
Yes
Yeah.
or no?
The blob.
Yeah, we're going to have to go shake the dirt off here.
Greg, are you okay with one of these?
He says 2 would be good.
Sounds like
It'll clear in a second.
Oh, he's in. Okay, he's in the jar.
Yeah.
Cool.
Yeah, you could quit
Yeah, it's done.
him.
All right, he's saying 1 is good now.
one is good.
1 is good.
All right.
Because of
we're-
all the dirt.
Yeah, I think we will be fine if we start moving towards
Okay.
our waypoint.
This will clear up relatively quickly here.
They only want one, Bob. Only want one.
Okay.
Sorry to disappoint you.
Two for one sail.
All right, can you
See? No.
can you sit here for a minute and then wait till you can see,
Yeah,
and then we'll talk.
but
We'll
as soon as we move anywhere, we're going to be
instead of-
buried.
Okay. Well, then we can talk while we.
All right. Well, look, if you can see right now to take the second sea cucumber, we will. I'm pretty sure it's not going to clog, but what we'd like to do is avoid spending a lot of time here.
Yeah.
Let me know when you want suction.
Do it.
Doing it.
Slurp.
And there he goes.
Join your pal.
Things are about to get tight in that jar number 7.
I know. Hopefully they were friends. That definitely was a different sea cucumber than any of the other ones we've been seeing.
I can cycle it.
Yep.
Yeah.
Okay. So will we count this as one sample just with 2 cucumbers?
Yeah.
Yep.
Okay.
Yeah, there he is.
There it is.
Go in there?
Yeah, that's
Yep, you
that's
got
Yep,
the
there's
boat.
2.
Lovely.
Good job. Thank you.
Cool.
So we're like really filthy, so I need to shake,
Okay, do a dance and then we talk.
wag the dog.
After you dance, we talk. If you want to dance over to this waypoint that you're very close to,
Which waypoint?
this AL. I'd like
That's right there.
Yep, I would like to see this little yellow box on top of that purple.
Really?
And then we're going to follow a very specific trajectory.
Well,
Do it.
how far are we going and what are we doing to get there?
We're going to go 053, about 204 meters to the next waypoint with, I'm sure, stops along the way, but we want to try and definitely cover that line because they picked it up specifically
Okay, you don't
over
need to
ridge.
look at what's there. It's
We
just right
we
there.
do need to look at the bottom once we get into out of
No,
this.
I mean this bottom's right here, 2 meters away from where we were at.
It was 6 meters. Don't you give me lip.
Aaron, is it possible to pull up that last target real quick?
Oh yeah, sure.
Thank you so much.
No,
Or, I don't know if it was the same as
Oh,
the others.
not gonna see who he is.
it was probably a little different location. Very small change.
Okay, so
That's
can I shake it off or
Yep,
what?
shake it. Well, wait, do you want to take any screen captures or anything here? He's right over that target.
No,
Okay,
no screen capture.
shake it.
Okay.
You got a couple of pictures of the bottom already.
I did. Yep.
I'm gonna be
Is that a
just coming up
Brazilian
and down.
At 9 o'clock.
Okay.
It's a
Just,
Brazilian.
you don't need to chase
Okay.
me around.
Thanks,
I don't
Aaron.
know. I'll just give
Just
you
gonna make a
some
big mess
space.
here.
I got that position, thank you.
Yep.
What I'm really hoping is that we hit some hard ground soon.
Yeah. I really like when we're far up, this white stars look like little twinkling.
All right, well, are we good?
I think so.
What's the bearing?
053. So get lined up on that and we'll get a ship move going.
There's your missing.
Actually, you come around the other way.
I'm going to call it in. It will take a while. Bridge nav. Can we get a 50 meter step bearing 053?
We're going right back to this. I guess we're just twirling to clean ourselves. Clean Herc. Is that what we're with?
Okay, 053.
Good morning, 8 to
Is
12
this
in
a
the van.
transect?
This is Samantha
What are we doing?
Wishnak calling in from shore.
Hi, Megan.
Good morning.
Good
Oh,
morning.
Samantha
Good
calling
morning.
in, actually.
Oh, Samantha. Hi, Samantha. Sorry.
No worries. I'll
Hey,
let you guys
Sam.
get settled and then bring in some viewer questions for you when you're ready.
Cool.
We're just getting lined up and get ready to start moving up slope. So minute or so and we should be settled in.
Sounds great.
This isn't a transect, this is just going.
No, unless, do you want to transect?
No, but when we get to the hard ground, we would like one.
Okay.
Take a picture of that.
And we may want to stop,
You're going,
really
right?
depending on what we
You
see.
going?
Yep, ship's moving. Yep. Just give us a shout. Are we happy?
Get
Everyone?
a
Front
jump.
row. Front row. Good.
Well, it's
Who,
all
me?
relative.
Are we doing the best we can? Coping. I think we're good, Sam. If you want to ask some questions, I think we're all ready.
What is that?
Alrighty.
Zoom in now.
please.
Sono buoy,
Sonar.
likely.
Third.
An old push core.
No, I'm betting sono buoy. We're like,
On a buoy.
yeah, we're like,
Military?
yeah, we're like right next to the range. So
Yep.
the range is on the just off of San Clemente. So We're in the-
Interesting.
we're in the bombing range right here, but we're close to the anti-submarine warfare range. So there's gobs of those.
So that was actually the first question coming in from our viewers, wondering where exactly we are and why is this area of interest to the team on board?
Well, we are at the base of the San Clemente Escarpment. We're about 44 nautical miles off San Diego and just southeast, maybe 20 miles from San Clemente Island, or maybe fewer. And this area is highly active tectonically. That means there's a lot of earthquakes and the earth moves and the plates move. And we are looking for ferromanganese crust, rocks that have a mineral precipitate that's of interest because it often contains a lot of metals that people are interested in. We are here to look at the biology and the microbiology and characterize the communities associated with those crusts. But at this exact moment, we're heading towards the steep escarpment. We're right at the base, and we're looking to see what this first little ridge of potentially of hard ground might be made of, just in case there's a methane seep here.
Ooh, exciting. Okay, so when you say associated communities, what kind of communities might we be finding around these areas?
Well, on the rock, oh, we just had a sea cucumber go by. So right now we're looking, we're looking at brittle stars and sea cucumbers
Cardboard?
on the sediment. Oh, let's take a look at these.
Corrugated cardboard.
I'm just wondering about the tubes. So, but during this dive series, we've been looking at rocks with corals, sponges, bryozoans, and then a lot of small creatures, small worms, and protozoans attached to them.
Can we zoom in again?
Cardboard?
I think it's unlikely that cardboard would be that pristine. It's probably a corrugated plastic.
Yep.
Or fiberglass or something.
Right now we're just looking at a piece of trash on the bottom.
And what are these long, spiny?
But I'm looking at the
worms?
whether the worms, which are Hylanesias potentially, so a little perplexed. Yep, Hylanesia. They are called quill worms. They look a little bit like quills are ancient writing instruments, like pencils. And these worms live on the surface of the sediment and drag their tubes around with them. Sometimes they head over to organic matter and pile themselves up. We've seen that in Costa Rica. So maybe there was something to eat associated with this plastic.
Or maybe it is cardboard. It's an awful, no.
The way it's tearing, it looks like cardboard, but
yeah.
Who knows?
It's in awfully good shape for cardboard on the bottom of the ocean.
Yeah.
See the head of the worm poking out the tube.
Oh, yeah.
Yep.
Oh, that's interesting.
Right there is the head of the worm. It's got appendages on it. Hylanesia, I think, is in a newfid.
You got more zoom out?
That's all I got.
It's pretty good.
So for those of you on the science chat, we're actually doing some communication and answering questions from Samantha. Samantha, tell us where you are and who's asking the questions, please.
Sure can. I'm actually on shore.
Okay, zoom
I
out.
was just on Nautilus
Moving on.
and returned home to Moss Landing, California, near Monterey Bay, or right in the middle of Monterey Bay. So I'm taking questions from the Nautilus Live website. So if you're watching the Nautilus live stream on any other channel or website than nautiluslive.org, come on over to nautiluslive.org and there's a question bar that you can type in your question, and I'm taking these questions and relaying them to our control van team for the next hour.
Great.
And one of the questions that has been coming in is about the 2 sea cucumbers that were just collected a few minutes ago. Why were these specimens identified to collect for sampling?
That's actually a question for Greg Rouse. We have some of our scientists who aren't on watch right now, but are typing in requests to us. They're on the elsewhere on the ship. I know these cucumbers will go into the Scripps benthic invertebrate collection and be archived. We do try to collect the really common taxa. I don't know, I'm assuming Greg asked for them because perhaps he doesn't already have those specimens in the collections. He called them warty sea cucumbers. I'm not sure whether Paul is sampling cucumbers for associated microbiomes, is he?
Yeah,
No?
we have already sampled the sea pig cucumber, and that was a very interesting one as well.
Now, can you give me a junk zoom?
So there's interest in the bacteria that are associated with these animals, sometimes in their guts, that maybe have special properties of interest. Greg said he didn't recognize this one, and again, Paul will use it for this species for extraction to look at the genomes of the microbes.
Very cool. Another question coming in. A viewer writes, a few days ago, there was talk about red animals being common in the deep sea. Why do sulfur yellow animals also seem to be common on this series of dives?
Could you repeat that a little bit slower? I didn't quite catch the whole question.
You zoom in?
Sure, I think my connection might be breaking up. A few days ago, there was talk about red animals being common in the deep sea. Why do sulfur yellow animals also seem to be common on this series of dives?
Why do what?
The yellow, the
yellow
really,
ones?
yeah,
sulfur
the really bright
colored?
one, yeah.
Well, we've seen some yellow sponges, and I'm not sure what that pigment is or why they have it.
Don't care about this guy.
We're looking at another cucumber here, different kind.
Okay, zoom out.
Can't tell if it's not a scotoplanes, something else.
Okay, you can zoom back in.
Does anybody else have a clue? I mean, you know, I don't know that much about the colors of the animals, but white is also very, very common. Deep sea, you know, basically no color. And we've seen a lot of those. We're a lot deeper now. I don't know if that's going to make a difference in terms of the colors of the organisms. That was a brisingid starfish that we just looked at. That's an asteroid, a different group of echinoderms than the brittle stars.
Greg is asking if we can slurp that sea cucumber right there by the brisingid.
Oh, all right. Before we go,
what is
could
that
we
thing?
have that pink cucumber, please?
There's a worm in the,
Scale worm?
not sure exactly, but that's fun.
Should have, okay, can you do that?
I don't know, Greg.
He said the next.
Patience, it's going to clear in seconds. I can see it in the front or down cam.
Oh
Okay.
yeah, that's true.
I think our watch will move on at 1 o'clock, so we get a little rock time.
Oh,
Is it still there? Or
Oh,
is it?
oh.
No. Okay, let's keep going,
Like poof,
please.
he's gone. Where did he go?
I don't know.
Huh. All right, moving on.
He doesn't know how
You
lucky
guys
he
gotta
is.
be quicker on the draw there.
That was a Halotherian, right?
That was,
Yeah.
yes.
And it
Okay.
ran away?
Let's head
Apparently.
over.
Well, we created it. We started to go and then stopped. So we created kind of a wave.
I'm just imagining a running Halotherian.
A running sea cucumber.
Yep.
Yeah. it's
Tennis
funny enough.
shoes. Lots of little tennis shoes.
We got our pile of dirt with us again.
I think the question about color is really interesting because all the brittle stars are white down here, and up in the oxygen minimum zone, they were often orange or reddish. I have no clue how those colors function. It might take less energy not to make pigments.
Zoom in.
Even the sea cucumbers are whitish here.
Maybe
Lots
a
of
sponge
miscellaneous
skeleton.
things here. Yeah.
Well, that's interesting.
Looks like corn on the cob.
I don't think that's a sponge. That's a
Yeah, too regular.
man-made thing.
Okay, let's keep going.
Keep going.
I'm liking this dive so
Moving on.
far. Even the sediment's giving us some cool stuff to check out.
One of the questions coming in from the viewers is what's kind of on the list for that the science team is looking out for on this dive? If there are geological targets or biological targets, other than clearly sea cucumbers are on the list.
Well, I think most of the invertebrates that are common here are on the list if they're not already in the collections or if there's a chance for microbes. Sponges, corals, biozoans, gorgonian corals, I should say, octocorals are on the list for the collections, but so are the rocks. And the Jensen lab scrapes the rocks and the crusts on the rocks for microbiological samples. And we pull all the other animals off.
Yeah, and on this dive specifically, we're hoping to find some nice outcrops with a mineral type that's called a ferromanganese crust, which is an iron and manganese oxide precipitate that forms on top of other kinds of rocks like volcanic basalts or sedimentary rocks where there is surface area available that doesn't have sediment on it. These ferromanganese crusts can precipitate on top of those rocks and create almost a secondary deposit on top. And these ferromanganese crusts grow about 1 to 3 millimeters per million years, so they're extremely slow growing, and they have a lot of electrons to share based on their chemistry, and so we're hoping that the biology associated with these ferromanganese crusts might be specialized to living in those iron and manganese rich oxide environments.
You planted.
And have you been finding these ferromanganese crusts on other dives during
Another
this
one of those
expedition?
sea cucumbers.
Yeah, we found, we got a really nice about 20 centimeter thick, or I'm sorry, 20 millimeter or 2 centimeter thick ferromanganese crust from San Juan Seamount, which was our furthest out dive. And we've also gotten some very thin ferromanganese crusts, or patinas, we call them, when they're about 1 millimeter or less, on some of the other banks that we've been looking at more inshore, especially northeast bank. But we actually mostly recovered phosphorites, the other mineral type that we're interested from the other sites, and we actually chose this dive today to go a bit deeper into some more rocky outcrops and rugged features to try to find ferromanganese crusts specifically on this dive. So we're hoping for a few more of those samples, and that's one reason that we're here today.
Starting to pick up some harder returns on the sonar.
That's good. I'm curious about the Brazinghids because they're not these starfish, upside down starfish with all the arms in the water are filter feeding, but they're usually a hard substrate animal that lives on rocks. So this is telling us that either there's some rock right underneath the sediment here, or there's something else going on with strong currents that I rarely see them sitting on sediment. So I'm guessing that we're on rock, actually.
Something happened with the delta. That's not right.
Yeah. So we're starting to pick up some gravel and small rocks.
It says we're only, delta is 3.4 meters. Yeah, hang on a second. Just hang right there.
Do you want to hold position to get it sorted out?
Something's amiss here.
All right, bridge nav, hold position.
We're heading
Arrow
up
was
over a
turned
little
off.
ridge.
How'd that happen?
We wanted to see what was here.
Yeah, viewers can still see
Okay.
the map on the quad view.
That's weird.
This is the background here is called slope, and the map is actually able to show the approximate slope.
Do you want to look at these rocks close up?
Nope.
Is there any
We
reason
can,
to?
I mean, we should be keeping our eyes out, but I'm hoping to find somewhere a little bit more outcrop attached and less sedimented for the ferromanganese crests. So in the map, we can see the slope, and we're getting to a more slopey area.
We are picking up animals now, sponges, hard substrate animals. Can't tell if that's an acesta or something else. Probably a sponge. And so now we're in some mixed sediment and rocks.
And so
Do
here,
you think you can slurp that sea cucumber?
Get over there.
That looks like another scotoplanes.
I'm sorry, we want to try and sample that one?
Yes, please.
Okay. Bridge nav, hold position.
Well, I think we need more over here, center screen.
and so a lot of these rocks
That's good.
look blackish, and that is likely a little bit of a manganese oxide coating or a very, very thin ferromanganese crust.
We'll want to use one of the coarse canisters, 4, 5, or 6.
Can we get a close-up before we disturb the sediment?
Yep.
Thank you.
Right now. Tilt up.
Wow.
Is this the same cucumber, Greg? It actually looks different to me.
It does.
And do you want it? Yes, please. It's not the scotoplanes. Somebody else.
And if you can see on the rock next to it, a little bit of a bumpy texture.
Synallactidae. It might dissolve.
Show me the hose. I need to see the slurp hose.
Do you want to run the flush?
Yeah.
Okay, I'll get that lined up.
Nailed it. Sweet. It's a sample jar dance.
Yeah. Ready for flush?
Yep.
Okay. Flushing is on. You good with that?
Yep.
All right, killing that. And then you get the jar rotated.
Which jar are we shooting for?
Give me one moment. I think it's gonna be. Okay, that's 1. Is that 5? Did I say 5?
Yeah, that's 5.
That's 5. The other one was 4.
Let's do 4.
Did you say 4?
Yes, it will be slurp jar 4.
You good?
Yep.
Not gonna be too big. Don't suck it too hard.
Okay, slow.
Just give it a little bit and we'll see what it looks like. So I might have to shake it out.
Greg is saying it might dissolve also.
Interesting.
Oh.
Well, give it the beans now.
All right.
On ascent, it might dissolve.
Oh, okay, on ascent, when the pressure changes.
Yeah.
Oh, I think
It's
he's
in
in
the
there.
jar.
Yep.
Which sample was that?
This is NA124 175.
175, roger.
Okay.
Greg is saying the collagen on the sea cucumber might just melt on ascent.
That's so interesting. Can we watch that jar on ascent? See if we see it go away.
Lots of questions coming in about the cucumbers. Are these different species that are being collected? And if so, are there different reasons to collect them, or will they be processed and analyzed in the same way?
They are different species.
It's pretty steep here.
Yeah.
This is a different species
keep
than
it
the
slow.
previous collection, and I think we've seen at least 4 sea cucumber species. One of them was like pelagic and floated by in the water, and we saw 3 different ones on the bottom.
Video bump zoom, please.
Yeah, this
Are you
spot
ready to continue on?
seems,
Thank you.
rich in
Okay, we're
sea
going to do
cucumbers.
some keep it 10
Diversity
meters
and
steps, if
numbers.
that's all right.
Yeah, that's all right.
Bridge nav.
What did you say, Gabe?
This site, what we've seen so far, looks pretty good in terms of
1
diversity
0
and,
meter step bearing 053.
diversity and number of sea cucumbers.
Oh, 4.
Yep. The swimming one we're seeing are the Enipniastes, and Greg is saying we don't need those.
This sponge is Voesilastra Ademospongiae.
Celeste.
I guess I would rover it.
Hmm?
Looks like we're kind of over it here. I think.
Over the slope?
Yeah. I don't know.
You think so?
Extensive. Is it supposed to go on for a while?
I think there's like one little kind of mini ridge,
It's ridge. Yeah.
and then it flattens out again, but we shouldn't quite be at the top of it yet. I'm going to keep this step small for the time being.
Yeah. Okay.
Bridge nav, another step, same bearing.
It's so funny to keep size in context. Like for a minute, I was thinking those were big large boulders, but that's the lasers that you can see on the screen are actually 10 centimeters apart. So these are actually individual rocks, large rocks, but not boulders.
Sponges have a lot of brittle stars on them. We don't always see that.
True.
Oh, is that a crinoid? We might have to get it for Greg. Can you slow down, please, and zoom in on the sponge with the crinoid and the clams on the wall there?
Trying.
All right, we have a request for both the crinoid and the
I
sponge.
think it's going to be too low to see it.
Can I get
Maybe
closer? My camera's
I
tilted.
think it's got some brittle stars on
Okay.
the sponge too.
You're good. Just plant it there. I think
Plant
We
it
don't
there?
have a request
Okay.
for those, but we'll get them anyway.
That's quite a pretty
So-
little
It's a whole
collection.
package.
Yeah,
Oh,
there's
no.
some Asusta.
I'm sorry, but it's you see what it is?
Yeah. I
Okay.
Just, that was. Yeah.
Sorry.
It's all right. It's all right. Just stay down there.
Greg, do we want to
Might be
suction
like up in the
these,
light bar.
or do you want us to try to put them in a
It's
bio
pretty-
box?
I'm gonna get closer.
The crinoid,
yeah.
for example, will it go
You have
up the
to
suction?
land on the rocks in order to get to it.
Okay, I can do it.
Okay.
You'll need to.
Just be patient with me, yeah.
Don't run into
So,
it.
Kira,
Yeah,
do
back
you
up.
think this is a ferromanganese coated rock?
Yeah, I definitely think there's a ferromanganese coating on the outside of the rocks. Could be possible to try and sample something from this large outcrop. And I think we're starting to see some more interesting biology as we get these large
Oh, the joy
rocky
gain's
features,
cranked
and
way up.
Yeah.
we have some information
It's
that
been
it looks
that
like
way.
this is
Okay, it's whatever you're used to. Just
the
100 is hard because it's.
So where the where the lasers are, just right in there, yeah. That it's very close, but that's the only place to
No,
park.
you got to turn and land over on the side of the rock.
To the left side.
So that side, yeah.
Okay.
So you got to turn right and lateral over there.
So
But see, you got way too much joy gain. It's
Okay.
just messing you up.
Okay.
There.
So we'd like to try to pull the sponge and the crinoid on top of it into one of the bio boxes.
Okay. Turn.
I changed my heading.
Yeah.
But we'd like to get some good photos first.
Yep.
Keep turning.
I'm turning. Thank you.
Right. So the bivalves, what look like bivalves, could be brachiopods there also. We need to take a
Closer.
Keep turning. Keep turning.
Okay.
Turning. Keep turning. We want to land on that face over there. So that. Oh, easy.
I just don't want to drive into it. You're making me nervous, Bob.
Well, you're making me nervous.
All right.
So tilt down a little bit
My
so
camera.
you can see where you're doing. Yeah. We want to put the basket up against the rock.
Okay.
We don't want to put the forehead against the rock. Tilt down more and just come up so you can see what you're doing. Okay, now come up.
Come up. Come up. I'm coming up.
Easy.
Okay. Forward a little. Plant it.
Well, keep coming up. You got to land the basket on it,
Okay.
not the forehead.
Okay.
You got to come up.
You want to do this?
Yeah.
Okay.
I'll
Cool.
show you.
Sorry. Thank you. I'll watch this one.
I'm curious, Lisa, how could you tell the difference between the crinoid and the brisingid?
Well, they're both echinoderms, but they're in different orders, I believe. They look different to me, of course.
Yeah.
You haven't had a good look at the crinoid yet. So let's get in there and you'll take a look, and it'll probably look quite different to you. They come in stalked and non-stalked forms. This is a non-stalked crinoid that I agree they do look a little bit like the brisingids, which are basically upside down.
We turn the porch light on?
starfish.
Sure.
It's not on.
Yeah, I had it
The
off
number of
before.
arms
Sorry.
might be different.
Okay.
Crinoids don't necessarily always have 5 arms. Maybe they have 10.
Two.
I think they
I'm counting
have
10.
I think they have pentagonal symmetry. Greg, Gabe would like to know what the defining feature between the brisingid asteroid and the crinoid is that we
Zoom in.
visually tell the difference. There we go. You can see they have structures that are holding onto the sponge. The
Yeah.
arms themselves look quite different.
That was hard.
Okay, good.
The coloration is often different. Crinoids can be many colors, but yellow and orange are common. Red. Brisingids don't have cirri, so these long tentacles there. Crinoids have the cirri to hold on. The mouth is upper, and in the brisingids, the mouth's actually on the bottom. They just live upside down.
So what's the plan here for this?
We would like to put the crinoid and the sponge together. I think we'll just start the forward bio box.
Is it going to hang on there, or is it going to swim away?
Well, if you put him, can you grab from with the crinoid in the claw?
Perhaps. It's pretty precarious here, but we'll give it a whirl. And where's it? Is it going to go on the front box?
Yeah, let's just put it on the front, one side or other of the front box. That'll be safest. Crinoids can swim, so it's possible it would swim away. Greg said don't grab the crinoid, just hope it goes with the sponge.
Okay.
So maybe don't touch it. You can avoid that.
Okay.
It's possible if we touch it, it'll swim off.
So they're not cemented on there.
We also have a request to zoom over, look at the shells, but I think we can do that after the sponges collected.
This is pretty precarious position here.
Yep. You touched him.
It was a friendly gesture.
High five.
It's a pretty sketchy position.
Oh,
Hang on, crinoid.
you can see it changed its demeanor.
It's playing dead.
It's feeling, trying to see what's there.
Can you pan me over?
Yeah. Left.
Yeah, so I could see.
It's a tough grab.
Nice sample.
It's not in a
Not
box.
in yet.
Rack
Don't
back.
jinx it.
Oh, ooh, don't move me yet.
Not touching.
It's gonna kick me off of this rock.
Okay.
Really great screen grabs of that.
It's beautiful.
Oh.
No.
Oh, no. Okay. What's the plan, Bob?
I gotta get it over the box there.
Can I open the box, or is
It's
that
gonna
gonna
kick
mess?
me
Yeah,
off.
yeah.
I'm just trying to get where I can hang on to it.
Okay.
Rack the camera back. Sorry. Okay. Box out gently.
Box out gentle.
It's gonna start to kick me off the rock. So, which side is it going in?
Either one.
Outside.
Red 5.
Okay.
More.
Yeah. Keep going.
No more.
Yeah. Keep going.
I'm going.
Are we counting that as one sample or two?
I think
There's
one sample. Yeah. Sample 176.
You want more?
I don't think so.
Oh, yeah. Okay. Hang on. Boy, this is freaking hard.
You want me to close the drawer? His
Nope.
no, he's sticking out. Yeah.
Make sure he doesn't swim away.
Okay. Close the drawer. Oh, got an arm.
His other arm's
didn't-
sticking out of the box on the other side.
Well, I don't want to
You want to push him in?
monkey with him. Yeah,
Okay,
just
close it.
Close it.
Tilt tray in. Sir, get your
Oh,
fingers
yeah. Go back out.
out.
He doesn't seem like he's really into swimming.
Don't say that.
I don't know.
That was 176.
So we had a request for more of the sponge. I don't know if that's going to be possible or not.
Is it okay if I close it and see if he just bends under?
Yeah, you can try it. See what happens.
Yeah, he looks.
Data logger, can you confirm which box that one is?
He's not moving.
Raj,
now.
that was 176. That was red 5.
Why don't you come out again?
Sure.
And what is this that we're collecting?
It's a crinoid and a sponge.
Yeah, it's two in my face here. Come out somewhere.
Out somewhere? Sure.
All right,
All set?
I guess.
Coming in.
We'd like to have a zoom on the
He just got his fingers
bivalves
stuck in
and
the car door.
another piece of sponge if possible.
Okay, can we zoom in?
Could I see your notes for the last one?
Which,
Uh,
where's the bivalve?
the one
Oh,
that
right
went into
there,
slurp 4?
up there.
Mm-hmm.
That guy?
Yeah.
All right.
Is that our max zoom?
That's good.
That's perfect. Thank
All
you.
right, so are these acesta? They don't look like the one we collected yesterday, but they don't, or are they brachiopods? They don't look like brachiopods to me. Greg says yes, they're acesta. Crispin tells us they're not brachiopods.
and we want another piece of sponge?
Yes, please.
Can we grab the piece of sponge that fell off, or do you want a different piece? You see it in Argus? It's a big chunk.
It's a big chunk?
Yeah, that, oh wait, is that?
That's another sponge.
Oh, sorry.
It's another sponge.
I
Sorry.
didn't grab a big
It
chunk.
almost looks
No.
like puzzle pieces.
So we're not gonna hurt that other sponge.
Cool.
We're going to continue.
Wait, what's on the basket?
Yeah,
Down
that,
at the basket.
the,
Piece of sponge right on top of the plate, Alvin plate.
there's some right there, yeah, on the Alvin plate.
That'd be hard to get. But it's probably.
What are you looking at?
There's a piece of sponge on the basket on the plate.
You can see it in the bubble cam.
Yeah, but I can't do that.
I'll help you, I'll help you. A little more starboard.
You're starboard or my starboard?
You're starboard, I'll talk to,
Oh, he's
I think you
got
can
it.
see it now.
See it now.
Same box?
I don't quite have it. You're outside the crate with your one claw.
Oh.
Is it like picking up a dime with a pair of
Yeah.
chopsticks?
I think we'll be fine. Yeah.
Devin, could you confirm the time for slurp 4 in the log?
Dang it. Get over there.
Can you repeat that?
That was for Devin.
Sorry?
Could you confirm the time for slurp 4?
I can't go back and look at times in the computer yet.
I have 1848.
I think that's too late.
You're probably right.
You got it.
We can check the computer later to confirm it. I just can't do that right now.
175 was 1833.